So far, we've been talking about the DNA present in the nucleus, and that is the DNA that contains our genetic information. But it turns out there's actually two kinds of DNA within our cell, because not only is there DNA within the nucleus, there is also DNA in what is called the mitochondria. And the mitochondria are present in the cell, and they're part of the energy system. And they contain their own DNA. And it is different to the nuclear DNA. The nuclear DNA is a long chain. The mitochondrial DNA is circular. It has 16,569 bases organized into 37 genes. It does have non-coding regions, and they do exhibit the kind of variation you need to distinguish individuals. The good thing about mitochondrial DNA, what makes it useful, is that it has a high copy number per cell. Within a single cell, there is only one copy of your nuclear DNA, but there are multiple copies of your mitochondrial DNA. It is also more robust than nuclear DNA. It's more resistant to sample degradation. So if you have a biological sample that's old or has been exposed to harsh treatment, then you're more likely to be able to extract and analyze the mitochondrial DNA compared to the nuclear DNA. There is a very important difference between mitochondrial and nuclear DNA, and that is mitochondrial DNA is inherited strictly from the mother, not the father. This means that mitochondrial DNA is not unique to an individual. A child will have the same mitochondrial DNA as its mother and the same as the grandmother, all the way up the maternal line. There's also faster mutation, generation to generation in the mitochondrial DNA, and that's because the proofreading is not so good compared to nuclear DNA. So, here's an example of the use of mitochondrial DNA for samples from which it's not possible to get nuclear DNA. And this is the case of the Romanovs, the imperial family of Russia. Now, following the Russian Revolution in 1917, the imperial family, the Romanov family, the Tsar, the Tsarina, and their children were detained, ultimately in the city of Ekaterinburg, and in 1918, on the orders of Lenin, they were murdered or executed, depending on your point of view. The Bolsheviks attempted to destroy the remains, and the bones of the royal family were eventually buried in the woods, and the Bolsheviks hoped that they would be forgotten. But they weren't, and after the fall of the communism and the fall of the Soviet Union, the bones were excavated from where they'd been hidden, and the Russian government decided to give the imperial family a proper funeral. But if you're going to organize a grand funeral for these human remains, you first have to make sure they really are the remains of the imperial family. It's not something where you want to have a mistake. That would be very embarrassing. Well, the remains have been burned and buried and all sorts, so of course it's not feasible to get nuclear DNA. But they were able to obtain mitochondrial DNA because that is more robust. So let's look at the imperial family. The father of the family, Tsar Nicholas II, some of his relatives survived the revolution. They escaped from Russia. Now, we can trace back the Tsar to his mother and to his grandmother, Louise of Hesse-Kassel. So Louise of Hesse-Kassel's daughter was the Tsar's mother. The Tsar has a sister. The sister had a daughter who had a daughter who had a daughter. So we have an intact maternal line, and all the female members of that line will pass down the mitochondrial DNA, which is the same as Tsar Nicholas's mitochondrial DNA. And one set of bones, those of an adult male, there was a match, showing those were the remains of the Tsar. Technically, you could argue they could be the remains of the Tsar's brother, but what happened to the Tsar's brother was known, so he can be eliminated from the investigation. Now, what about the other remains? The 4 daughters of the Tsar and his one son, Alexei. Well, they won't have the same mitochondrial DNA as the Tsar. They will have the same mitochondrial DNA as their mother, the Tsarina Alexandra. The Tsarina Alexandra had a sister who had a daughter who had a son. And that son was Prince Philip, Duke of Edinburgh, husband of the Queen of England. Prince Philip, therefore, should have the same mitochondrial DNA as the Tsarina Alexandra. So he donated a sample of DNA, and sure enough, there's a match of the mitochondrial DNA with the mitochondrial DNA of the remains of the woman and the children. So this analysis confirmed that the remains found in that forest near Ekaterinburg were indeed those of the ill-fated Romanov family. Well, while we're talking about the Romanovs, soon after the murder, stories went around that one member of the family, the Grand Duchess Anastasia, one of the daughters, had escaped and survived. And a woman appeared in Berlin in the 1920s claiming to be the Grand Duchess Anastasia. And certainly she had some physical resemblances to the Grand Duchess, and some members of the imperial family thought she was speaking the truth, and others thought she was a hoax. Well, ultimately, this woman emigrated to America, married an American, became Anna Anderson, and died in the state of Virginia in the United States in 1986. Sometime after, she was subjected to a DNA analysis. Now, when she died, her body had been cremated, but at some point she had had a medical investigation and there had been a biopsy, and the tissue taken for that biopsy had been preserved. So it was possible to extract DNA from that biopsy sample, and it proved that she was not the Grand Duchess Anastasia. She was actually a Polish lady called Franziska. Even so, even with the DNA evidence, that doesn't stop movie companies making a movie about it. Well, so far, we've talked about the DNA of human beings. But all species have DNA. So in principle, you could do DNA profiling of any species if that living organism was involved in a crime. So one excellent example is the Maricopa case from Arizona. And it dates back to 1992. A woman was murdered and her body was dumped at an abandoned factory in Maricopa County in Arizona. Now, based on ordinary police work, a suspect was apprehended, but he denied that he had ever been to the site where the body was found. Now, this suspect drove a truck, and when his truck was investigated, they found in it seed pods from a local tree called the Palo Verde tree. So the police suggested that maybe he had been at the crime scene because there was a Palo Verde tree there. Maybe he'd parked his truck under the tree and the seed pods had dropped from the tree into his truck. However, there's more than one Palo Verde tree in Arizona. So how do you prove that the seed pods in the truck came from the Palo Verde tree at the crime scene? And the answer was DNA. First, the forensic scientists had to collect DNA samples from many different Palo Verde trees and proved that the technique they used showed they could distinguish these individual trees. So they had to show there was variability and that they were detecting the variability. That they did. And then when they compared the DNA from the seeds in the truck to the DNA of the tree at the crime scene, there was a match. And the suspect was subsequently convicted of this murder. So we'll conclude this lecture with the case of Peter Falconio and Joanne Leese. And they were a couple who were touring Australia back in 2001, and they got themselves a camper van and they were driving around Australia. And their journey took them through the very sparsely inhabited Northern Territory. And as they were driving along through the Northern Territory, they were stopped by the driver of another vehicle. And the man in the other vehicle went off out of Joanne's sight with Peter. She then heard a shot. From that moment, Peter Falconio has never been seen again. Joanne was captured by the man and her hands were tied together with a strap. But she was able to escape when he was distracted and hid in the bush. He searched for her, didn't find her, and then he drove off. When she felt safe, she came out of the bush, was picked up by a truck driver, and taken to the nearest police station. When the police investigated, they were never able to find Peter Falconio's body. They did find one bloodstain on the road. Shortly afterwards, a suspect was arrested, and he was a drug smuggler. His business was smuggling amphetamines across Australia, and his name was Bradley Murdoch. But the question is, could he be linked to the crime scene? There were no witnesses except for Joanne Leese, so it was his word against her word. Now, on her t-shirt, there was blood. The DNA of that blood matched Bradley Murdoch. And the Australian forensic scientists calculated these tremendous odds. And when you look at these odds, you realize that blood must have been Bradley Murdoch's. But the blood stain was on her t-shirt. It was not at the crime scene. So it only proves that Bradley Murdoch had come into contact with Joanne Leese. And it does not prove they came into contact at the crime scene. It could have been at a gas station or something hundreds of miles away. But they were able to extract DNA from the gear stick of the camper van. And that DNA matched Bradley Murdoch. This puts Bradley Murdoch at the crime scene because it proves he was in Peter and Joanne's camper van. And then the third piece of evidence is the strap that was used to tie Joanne Leese's hands. There was not much DNA that could be extracted from it, so they had to rely on low copy number DNA. And they were able to extract from it the DNA of Joanne Leese. It had been tied around her wrists, so obviously it would have her DNA. They were able to extract from it the DNA of a man called Dr. Peter Thatcher. Well, who was he? Well, he was one of the forensic scientists, and his DNA had contaminated this sample. And finally, they were able to extract DNA of Bradley Murdoch from the strap. But this is low copy number DNA. So the probabilities that they calculated were a lot lower than for the bloodstain on the t-shirt. When I say a lot lower, they calculated a mere 100 million to 1. But given that the population of Australia is about 20 million, that's pretty good odds. So, what do we have? We have the blood on her t-shirt. If we did not have the DNA technology, how valuable would that evidence be? Well, without DNA technology, you could only use blood type. And if he'd had a rare blood type, you might have got 30 to 1 odds. What about the gear stick of the camper van? Well, without DNA technology, you wouldn't have got any evidence of that gear stick. Nothing. What about the strap? Again, the only thing linking the strap to Bradley Murdoch is the DNA. So without DNA technology, there's no evidence. This is a statement made by one of the prosecutors at the trial, who said one of these things by itself might not have been enough, but together there was a very damning case. So Bradley Murdoch was sentenced to 28 years, at least 28 years in prison, whereas without DNA technology, there would have been no case at all. So to sum up DNA, you've seen how we can use DNA technology to prove paternity and maternity. You've seen how DNA technology can facilitate police work, and that something as small as just 9 cells can be a trace that can be individualized. And you've seen that combining DNA technology with the use of DNA databases has allowed the solving of many, many cold cases involving both the conviction of the criminals and the freeing of the innocent.